Now let's study the Human Microbiome Project. Human Microbiome Project. Human Microbiome Project. See, this project, like as the research is going on, scientists are thinking that these microbiomes, they are very, useful for us. Okay, very useful for us, but they don't have any, the exact database. Fine, exact, the basis, exact, the information. They won't have the exact characteristics of those organisms which are involved in the normal flora. Understood? So the US, one institute, United States one institute, US institute called it NIH, okay, National Institute of Health. They launched this Human Microbiome Project in 2007. Okay, they launched the project in 2007. Fine. In short, they are also called at HMB. Human Microbiome Project. Fine, Human Microbiome Project. So it is United States, institute that called as the National Institute of Health. They launched in 2007 this Human Microbiome Project, and the aim was to first characterize the microbiomes, or the microflora, normal flora. Fine, characterize all the microflora. Understood? Then understand their physiology. Okay, understand the mechanism, the way by which they are interacting with us. Fine, their interaction with host, the interaction with host. Then understand how they are useful for us. Okay, their beneficiality, their benefits. Understood? Then how they are, associated with the diseases. Fine, how they are associated with the diseases. Fine, understood? So these are some of the, aim of launching this. I must say it's a wonderful project. Yeah. So this is launched by the United States agency called NIH, National Institute of Health, launched in 2007. The first aim is to characterize this microflora. Okay. Then understand their physiology. Understood? Then how they are interacting with the host, how they are they are considered as a beneficial for us, and how they are associated in the disease processes. Okay. These are some of the aim, but the higher aim is, diverse. There is initiative for, developing this large database, okay, to catalog all the, characteristics of these microorganisms. Understood? Fine. Along with that, it is involved in the understanding of the microgenomics, means their this flora genetics, okay, their flora genetics, they are also involved in their flora genetics. Now the second things which we have to keep in our mind about this project is that these projects are in two, you can say, stage first, or you can say in the two phase, okay, phase 1 and phase 2, okay, they are launched in the two phases, phase 1 and phase 2. Fine. In phase 1 is launched in 2007, okay, it is launched in the 2007 and it is completed in 2014. Yeah, I'm right, it's 2014. It is started in 2007, completed in 2014, the 7-year work, okay. And you know the cost, 170 million dollar. Fine. 170 million dollar they are getting the fund. They are getting the fund, 170 million dollar for developing this project, okay. To you can say completion of this project, this much amount is 170 million dollar. 170 million dollar. Now the first phase, understood, the first phase M is focus on identifying and characterizing the human microbiota, okay. The first phase M is identification, okay, and characterization of human microbiota, the organism which are, you can say, present in us, like the beneficial organism. Understood? No problem. Now in the second phase, this second phase is referred as a integrated human genome project, okay, integrated human genome project, integrative human genome project. It is started in 2014. Fine, it is started in 2014 and completed at 2016, okay. And as I said, it is also called as, yeah, it is also called as IHMP, integrative human microbiome project. Write it down, integrative human microbiome project. Understood? And the M of this second phase is that it generally, you can say, generating the resources to categorize the microbes, fine, and enlisting the data, the role of microbiome in health and disease, okay. Their M is to, you can say, understand, fine, the how these microbiomes are involved in the health, maintenance of the health, and in disease. Are you getting my points? Fine, understood. So this is about the Human Microbiome Project that is launched by the United States agency called at NIH, National Institute of Health, in 2007, okay. And the amount of the fund which they are getting is $170 million, okay. The M is to understand the microgenomics of the normal flora, to characterize the microflora, fine, to identify the microflora, understand their physiology, how they are interacting with the host, okay, ecological interaction, then their benefit and associate, how they are, you can say, associated with the disease. For that purpose, they are launched in the two phases, understood. The first phase started in 2007 to 2014, and the M was to identify and characterize the microorganism. Now the second phase, they are started in the 2014 and completed at 2016, fine. And their aim is first to develop tools, okay, in order to characterize or identify this microbiota which is present in the humans, fine. For that, the tools is needed, fine, the tools is needed. Then how they are involved in the health and disease process, this is the second phase, M. Understand? In 2016 this project is completed. Now what things you can say getting by this project, what promises you can say is done by this Human Microbiome Project. See this tiny organism and this much expenditure on just understanding the way how they are interacting with us. Now, so we will know first this as the project, what are the things they are finding. The first thing they find it is that this microflora or microbiota, human microbiota, they are beneficial for us. They are beneficial for us. Understood? Then it also enlisted some of the diseases like IBD, inflammatory bowel disorder. Okay, it's a kind of autoimmune disease. Autoimmune disease. Fine, understood? And here there is a, you can say, problem which we will discuss later. This represented by the two diseases. The first disease is called as the first disease represent the ulcerative colitis. Ulcerative colitis, fine. And the second one is called as the Crohn's disease. Crohn's disease, fine. And these are basically the inflammation of intestine, esophagus, fine. And it's the autoimmune diseases. In this disease, there is the involvement of this microflora. Okay, there is a, you can say, problem in microflora. Understood? So, and the another thing which we understood by the Human Microbiome Project is that, you know what, like the genes of this microbiome as compared to us, they are more in number and they are more functional in us as compared to our own native genes. Fine, their genes are more active in us and they are higher in number. It is surprising. No, one surprise we get it from the Human Microbiome Project is that the first surprise I can say which we are getting is that the genes or the microbes for genes of these microbiomes or microbiota, okay, they are more functional, fine. They are, you can say, more in number as compared to human genes or human you can say the native genes. Are you getting the points? Fine. Then it shows the relationship of pregnancy, okay, and you can say preterm birth. Preterm birth. Fine. How these microbiomes, they are, you can say, changing, fine, during the gestation period, during the pregnancy period. Fine, no problem. Now, the another thing it shows is that the microbiome, as I said, that microbiome contribute more genes responsible for human survival. Mark the words, human survival. They contributed for that more genes, okay, as compared to human own genes. Understood? The second thing is, it is estimated that the bacterial protein coating gene are 360 times more abundant than human genes, as I said. And there is estimation, you can write it down. How much time? 360 times. 360 times, fine. 360 times the bacterial protein coded genes are more abundant as compared to our normal genes, our normal native genes. This is the second surprise. The third surprise is the component of the human microbiome, okay, change over a time. Means the microbiomes which we have, it is subject to change over a period of time. Means they are also not a constant. because we know that we acquire this from majorly from the environment. Fine, it means it is continuous, subject to change over a period of time. It shows, you can say, the changes. It shows the changes. In order to survive, everyone have to adapt, no? So it shows the changing. Fine. Now normal condition they are changing, that's okay, fine for us, but in the disease condition they are dramatically changing. Okay, their behavior is changing for us during the disease time, disease condition. Understand? And once the disease is finished, okay, disease is, you can say, there is a cure from the disease, then they will again come in their, you can say, the normal behavior. So these are the three surprises, okay, which we get from this Human Microbiome Project. The first one is it is a beneficial for us. Second is it's involved in the many diseases. One of the diseases I shown you, that's called the IBD, fine, inflammatory bowel disorder. It's an autoimmune kind of disorder. It's represented in the two way, manifested in the two way. First one is ulcerative colitis, okay, and second one is the Crohn's disease, which we will discuss ahead later on. And the second surprise it shows is that the genes of microbiomes, the protein coding genes of microbiomes are 360 times more than the human genes. Understood? And second thing is that their contribution is far more greater, okay, than our own gene as far as our survival is concerned. Understood? They are also involved in the diabetes type 2 onset. Fine, understood, no problem. So now let's quickly conclude our class. What we studied that we cannot live isolated, okay, merely by the thinking, because we have certain, you can say, the microorganisms which are living in or on us, okay, in or on us. If it is living in us, we are referring as the endoparasite, fine, and they are living on us, we are referring as the exoparasite, fine. And this endoparasite, exoparasite, they represent, you can say, a whole classes of organism, okay, which is not a harmful for us normally, that are called as the microbiome, also called as microflora, fine, also called as the human microbiota, understood, and also called as the normal flora. So these are the alternative names. Remember, parasite doesn't mean that this microbiota or microorganisms. Parasite can be the another exoparasites, fine. Exoparasite means exogenous origin parasite, which are not you can say living in us naturally. Those parasites can cause a disease in us. Understood? Fine, no problem. Then we understood that they have the beneficial role. They are, you can say, they are performing a beneficial role for us. Understood? And sometimes due to their, you can say, the displacement, their dislocation during the disease condition, fine, they can cause a problem for us. No problem. Then we seen about their classification. They are classified into the two category, fine. The first category we seen is called as autochthonous flora, means the native flora, and another one is called as the allochthonous flora. Then we seen the distribution, how they are distributed. We made one conclusion. We seen that those parts which are actively, you can say, interacting with the environment, where they are living more, fine. And those our internal organs like spleen, kidney, liver, they are sterile organs, fine. There these organisms are generally not found unless until they are displaced from their natural location. Then we seen, you can say, the amazing project that we referred as the Human Biome Project, fine. We seen the aims of this project. We seen that the NIH, US agency, National Institute of Health, launched in 2007. We seen, you can say, the phases. They are in two phases, fine. We also seen about the outcome, the result from the promises, the surprise from this Human Microbiome project. Understood? So from the further class, okay, in the next class we will see about the names of organisms, the names of normal flora which is distributed in our body, in the skin, how many kinds of the names of, you can say, the organism which is present normally in the normal flora, what are their function, fine, and if they become, you can say, pathogenic, then what things they can cause. Understood? No problem. So thank you with having me. Bye. Have a nice day.